champions, so you have your biology exam today. Please don't worry about anything. You're going to do so good. Okay, I'm praying for you continuously. Lots of love from Jasmine ma'am. And do not panic. Draw the diagrams properly, underline important terms and write in points. That's all. Yeah, and please practice pedigree chart as well. I have not involved it over here because the time is really less, right? But you can practice it and revise carefully, and that's all. Let's go ahead. DS of anther or structure of microsporangium. So over here inside you have sporogenous tissue. To provide nourishment you have tapetum. Outside that you have middle layer. Outside that you have endothecium. Outside that you have epidermis. At least these many things should be there. Going ahead to the next thing, that is the megasporangium or the VS of anatropous ovule. Anatropous ovule means it's ulta. So usually we have a placenta, okay, which is there in the ovarian cavity, and this placenta is connected to the ovule with the help of a funicle. But this time instead of placenta being here, it's here, ulta, okay. And what happens? The place where it attaches, it is known as hilum. The ovule, it has outer inner integument. Inside the ovule, basically you have nucleus cells. Inside the nucleus cells, you would be having the embryo sac. This end is known as chalazial end. Wherever the egg is there, it is known as micropylar end. Okay. Going ahead, children, if you feel that speed is too much, please make sure you are putting down the speed. This is a setting, okay. You can lower down the speed if you want, but as this video has to be completed within 30 minutes, I have to speed up a bit, and this is a revision video. Just think of me at that friend who just makes you revise everything really fast at the start of the exam, right? Now, female gametophyte or embryo sac or 7-cell 8-nucleate embryo sac, my dear children, how do you make it over here? 1, 2, 3, 1, 2, 3, 6. Okay, 3 plus 3, 6, and then this whole thing is 7th cell, my dear children. If you talk about the nucleus, 1, 2, 3, 4, 5, 6, and 7 and 8. 8 nucleus is there. Okay, so all the cells are haploid in nature except central cell, which has 2 nuclei, which is why it is diploid in nature. Okay, or basically you cannot call it diploid. This nucleus is haploid, this nucleus is haploid anyways, okay. So at least you should be having this in your head, and wherever the egg apparate is there, there you have the micropylar end. So from the micropylar end, if you are asked to draw it properly, so first comes synergis, then the egg cell, then the central cell, and then the antipodals. Now going ahead, define polyembryony. Polyembryony means there are multiple embryos inside the ovule. Where do you find it? In mango and citrus fruits. Now this is an extension of apomixis, where basically you are making the seed or embryo without the need of fertilization, my dear children. Okay, where do you use it? What is the significance of it? Significance, advantageous, embryos are used to provide genetically uniform seedlings in the fruit and the trees. Yes, the polyembryos, the multiple embryos are all same, my dear children. So if it's all same and if it is all having good character, it is really good for basically growing some uniform seedlings. And apart from that, it is there for breeding and horticulture as well. This point you can avoid. Cool? Going ahead to the next thing, briefly explain the structure of pollen grain. So pollen grain, if you see, it is a pollen grain, and the pollen grain, my dear children, it has 2 cells. The bigger one is known as vegetative cell, the smaller one is known as generative cell. The bigger one has an irregular nucleus, the smaller one has a spindle shaped nucleus. It has a plasma membrane. Outside that it has a intine. It's intine. Intine is made up of cellulose and pectin. Outside that there is an irregular thing which is exine, and exine is made up of very resistant substance which is known as sporopollenin. And in between the spaces that you see between the exine, it is known as germ pores through which the pollen tube can be formed. Okay, so at least these things you should be having in your head, and 25 to 50 micrometers is the diameter of that particular thing. Works? And in 60% of the plants, my dear children, it is it is going to go in this particular stage, that is 2-cell stage with generative and the vegetative cell. Okay, in 40% of them, this generative cell is going to divide into 2 and make 2 gametes as well. So 1, 2 and 3-cell stage it will be going. Always remember, a pollen grain will be having 2 male gametes. It will be producing 2 male gametes. Now going ahead, we have what are the clistogamous flowers, right? 4 significance. Clistogamous flowers are the closed flowers which do not open at all, and it has the male and the female parts. It has the male and the female parts, so obviously every time fertilization or basically pollination would be happening for sure. So autogamy is for sure happening over here. Significance, clistogamous flowers are invariably autogamous as there is no chance of cross pollination, right? And clistogamous flowers produce assured seed set. Even in the absence of pollinators, they do not need pollinators, my dear children. There is no need of pollinators. Example of clistogamous flowers, Camelina, Viola, Oxalis. This is common pansy. Okay, remember they have 2 types of flowers. One is clistogamous and the other one is chasmogamous, the open one. The open one show both autogamy and geitonogamy. The closed one shows only autogamy. Okay, autogamy, geitonogamy, and also xenogamy if there is another plant. Okay, now What is pollination? Pollination is basically landing of anther on the stigma of the flower, right? Now, basically, if you're talking about wind pollinated flowers, what are the features that you should be knowing? They are small. The pollen grains are small, light and non-sticky. If they are sticky, they will go stick somewhere, yeah. And if you talk about the stigma, it is large and feathery to basically trap the pollen. Now, the stamen is just well exposed, my dear children. Why? Because the wind should be carrying it, right? The pollen grains. Now, no need for nectar or scent because there is no need of pollinators, right? Example is maize and wheat, and wind pollination is usually seen in the case of grasses. In the case of grass, and over here I can reckon, right, corn cob as well. This picture is of corn cob. This picture is this thing that you see over here is the stigma and the style that you have. The tassels are nothing but the stigma and the style. Okay. Now, which are what? Large and feathery. Now, over here, if you talk about insect pollinated flowers, this picture can be given to you, and you can write they are large, brightly colored and fragrant flowers, nectar present to attract what? Insects. They are sticky pollen grains for attachment to insect bodies, yeah, so that they can move around easily. Stigma is small and sticky for pollen reception. Example, sunflower, hibiscus, or whatever you want to write, you can write. Okay. Please go ahead, read by yourself about the water pollinated flowers, that is Vallisneria and Zoostera as well. Okay. Now, I hope I am able to make you remind that, yeah. And outbreeding devices as in something that does not allow inbreeding to happen, something that will allow cross pollination to happen, my dear children. So, what are those devices? One is production of unisexual flowers. If they are unisexual, obviously the flowers will be going from one plant to another. That will be better way, yeah. That will help in what? Listen, unisexual flowers can be on same plant also, where this is the female and this is the male. Both are unisexual, but from here to here it's happening anyway. Genetic makeup is not changed. So, this is an example of monoecious plants, my dear children, where gynoigamy is still happening. But if they are dioecious plants, where you have two different plants producing two different sex, okay, for example, this is the female one and this is producing the male one. So, over here xenogamy is for sure happening. So, production of unisexual flowers, apart from that, self-sterility or self-incompatibility, where pollen grains of a flower do not germinate on the stigma of the same flower due to what? Presence of some genetic mechanism. Okay. Now, you don't need to learn all this thing. Dichogamy is basically stigma and anther are not maturing at the same time. Okay. And then you have hercogamy. The term is not given in the NCERT, but just understand the natural physical barrier prevents the pollen of the same flower entering the ovary. That is not there. If you don't want to learn, no, it's okay. Okay, at least these three things you should be knowing. Now, going ahead, we have write the events of development of female gametophyte in order, or describe the process of megasporogenesis with the neat label diagram. So, this is the picture. First of all, plus understand. Okay, in the cells of the nucleus, one cell will become the megaspore mother cell. This megaspore mother cell is only going to undergo what? Meiotic division. Meiosis 1 enters. Okay, you have got two cells. Okay, you have got two cells. Now, these two cells again will be dividing because they have to undergo meiosis number 2, right? The meiosis happens in 2 stages. At the end, they will get 4 cells, 4 megaspores. Out of this, 3 will be degenerating. 1 will be the functional megaspore, which will be giving rise to the, embryo sac. Now, in the embryo sac, the nucleus will be dividing thrice. Mitotically, the nucleus will be dividing thrice, my dear children. Once it divides, you get 2 nucleus. Another time you divide, again you get 2 here, 2 here. So, 4 nucleus. Again, when you divide, you will be getting at the end 8 nucleus, my dear children. 8 nucleus will be getting. Out of them, 3 of them will be surrounded by cell wall. Here also 3 of them will be surrounded by cell wall. 1 of them is not surrounded. 2 of the nucleus are not surrounded by cell wall over here, my dear children. So, it is a part of central cell. This is how things happen. Okay, I hope it's clear. Please remember egg apparatus, filiform apparatus and everything out there. And over here, micropilar end is towards the egg apparatus. Going ahead to the next thing, what is emasculation? Emasculation is basically the removal of anther from the flower before they dehyse pollen grains. Okay, so because we do not want cell pollination to happen. So, we do it. We cut off the anther from a bisexual flower when there is both the sexes together. Okay. Now, differentiate between gynoigamy and xenogamy. Gynoigamy means you have one plant, 2 flowers. So, pollen grains, if it here to here, it goes anyways, genetically it is same. Xenogamy means one plant, another plant. 2 genetically different plants are being involved. So, gynoigamy transfer of pollen grains from anther of a flower to the stigma of another flower born on the same plant. It is functionally cross-pollination and requires pollinating agent, but genetically functionally cross-pollination, but genetically what? Similar to autogamy. Over here, xenogamy, pollination takes place between 2 flowers born on different plants of the same species, introduces what? Genetic variation. So, obviously over here, you will find more genetic variation. What is apomixis? The formation of seeds without fertilization is termed as what? Apomixis. Where do you see apomixis, my dear children? In the case of grass, Asteraceae and all of this thing. Okay, where what is happening? Without fertilization, seed is being formed. Without fertilization, seed is being formed, very much important. What is the importance of apomixis in crop breeding program is also being asked. Now, Define parthenogenesis. Parthenogenesis is different from parthenocarpy. What do you mean by parthenocarpy, my dear children? Parthenocarpy, apomixis, if you go ahead, parthenocarpy, here it is formation of seed without fertilization. Formation of fruit without fertilization is parthenocarpy. You can actually see them in banana as well. You have false fruit, true fruit. False fruit, the one that arises from other parts apart from the ovary, my dear children. For example, thalamus and things like that. Okay, example you can give of apple, strawberry and things. Okay, cashew nut. Now going ahead with true fruits, obviously they are going to arise from the ovary itself. There are certain hormones which level changes, which is why an ovary is getting into a fruit without fertilization. Please remember that as well. Going ahead, we have parthenogenesis. Parthenogenesis is found in haplodiploid sex determination, right? What happens? A haploid, cell is basically without fertilization getting to be a male. This is known as parthenogenesis. Formation of a new organism without fertilization is known as parthenogenesis. Example you find in honeybees, haplodiploid sex determination. Define embryogenesis, the process of development of what? Embryo. From the zygote, the whole process is known as embryogenesis. Now going ahead, distinguish between homogametes and heterogametes. Homogametes means they are going to produce same type of gametes, XX. Females, in human females, they are going to produce always X gametes. XY, the males, my dear children, they are going to produce either X or Y. So when two gametes are so similar in appearance, that is not possible to categorize them into male and female gametes. They are homogametes. When the gametes produced out of two morphological different times, they are known as heterogametes. Okay. Now differentiate between menstrual cycle and estrous cycle. The cyclic changes that occurs in primate, my dear children, okay, such as mammals, that is known as menstrual cycle. Monkeys, apes are the example of this. Apes, okay, not apes. Now, estrous cycle, the cyclic changes during reproduction in non-primates such as what? Cows, sheep and dogs and etc. It is what? Estrous cycle. Fine. Now, over here I missed something. Yeah, where do you find heterogametes, my dear children? So, in insects you find heterogametes. XO type of sex determination is there, where either X or no chromosome will be there. So, two type of gametes are there. Yeah, and in the case of birds also, in the case of Drosophila melanogaster also, they have XX, XY type of sex determination. Same, male show heterogamety. Female show heterogamety only once, and that is ZW, ZZ type of sex determination found in birds, my dear children, where they show female heterogamety, not homogamety. This is heterogamety. Okay. Now going ahead, we have female reproductive system. Very important diagram. Please go through it, all the layers and every single thing you should be knowing about thing. Now going ahead, male reproductive system, again the testes, the accessory ducts, the glands, the external genital organ, everything should be in your head. Going ahead, name the cells that secrete androgens. It is Leydig cells or interstitial cells that secrete androgen, and the most famous androgen is what? It is testosterone. Going ahead, what is menopause? Menopause means cessation or stoppage of menstrual cycle or the reproductive cycle. It is known as menopause. It happens almost around 50 years of age. What is menarche? The first menstruation. Okay, it starts in puberty and it is known as menarche. Now name the hormone secreted by corpus luteum. Corpus luteum majorly secretes large amount of progesterone, which helps in maintenance of what? Endometrium. It also releases estrogen, but progesterone is released way too much. Okay. Now, what is ovulation? Ovulation is basically the rupture of Graafian follicle to release what? The secondary oocyte, my dear children. Not the ovum, actually secondary oocyte, and this is known as ovulation. When does it happen? When the LH surge happens, LH increases way too much, and this phase is known as ovulatory phase. Going ahead, what is colostrum? Colostrum is the first breast milk the mother produces, and it has high amount of IgA. So what is the milk? What is the antibody that is transferred via the mother to the baby? It is IgA. Okay, via placenta to the baby, it's IgG. Mention the function of Sertoli cells. Sertoli cells, they provide nourishment. Okay, they provide nourishment to the spermatogonia which are there, the germ cells which are there. Apart from it, basically understand that Sertoli cells are going to release some factors, ABP, okay, androgen binding protein factors, which are very much important for the process of spermiogenesis. That is, spermiogenesis means the formation of, the conversion of spermatids to spermatozoa, the mature one. Okay, this is known as spermiogenesis. For this, you need androgen binding proteins, my dear children, very important. Okay, so this is being produced. Basically, FSH, follicle stimulating hormone, acts on Sertoli cells to produce this, and it goes and helps in this. It basically makes the testosterone stay up over here so that it can aid in this particular process. Going ahead with the next thing, mammalian mammary gland, very much important. Please know the flow. Mammary law first, then alveoli, then tubules, then ducts, then ampulla, then lactiferous ducts. You have so much fat, you have ribs and everything at the back. Okay. Going ahead, we have defined placenta, right? The function of placenta. Placenta is what? Temporary association. It's not there always, right? Between the fetal tissue and also the maternal tissue. From the fetal tissue, you have chorionic villi. From the maternal tissue, you have the uterine endometrium, my dear children. Okay, what does it do? Basically, it removes CO2, removes the waste materials. It is helping in the transport of oxygen and nutrient as well. It is also acting as an endocrine gland. 3 functions are there. What are the hormones? 4 hormones are there. Human chorionic gonadotropin, okay, human placental lactogen, estrogen and progesterone. Human chorionic gonadotropin is the one that maintains corpus luteum during pregnancy. Corpus luteum is important for the production of progesterone, which helps in maintaining the endometrium lining. Yeah, please remember, relaxin is not a placental hormone. Relaxin is an ovarian hormone produced in the later phases of pregnancy. Apart from it, there are 3 hormones whose amount increases many folds, okay, during pregnancy. One is cortisol, other is thyroxine, the other one is prolactin. 3 hormones. Done. Going ahead, describe the intrauterine devices. Intrauterine devices are the ones that is put inside the uterus with the help of doctors, and it's very important for spacing the children, as well as is one of the most widely accepted contraceptive technique. Okay, so you have 3 non-medicated, that is lipase loop, copper leasing, that is CUT and CU7, multiload 375, hormone leasing, that is progesterone as LNG20, my dear children. Non-medicated ones are basically going to increase the phagocytosis of sperm. So it is just something foreign, and when something foreign comes, our white blood cells become active. So anything that comes along with it, my dear children, whether it be sperm, they are going to be killed. Cu ions basically are going to mess up around the motility and the fertilizing capacity of the sperm. So they won't be able to move. And hormone releasing IUD such as progesterone or LNG20 basically are going to make the uterus unsuitable for implantation. So usually it does have progesterone, okay, but the thing is, when hormonal imbalance happen, usually progesterone does a good work, but when hormonal imbalance happen at this point of time, it will do the opposite. So it won't allow implantation to happen. It will make the cervix hostile to sperm by creating a mucus plug over there. So the sperm cannot transfer, okay, and also ovulation would not be happening over here. Going ahead to the next thing, artificial insemination technique. The semen collected from the husband or a healthy donor is artificially introduced into the vagina or the uterus, okay. This is known as, this is a part intrauterine insemination of the female. So intrauterine insemination is a part of artificial insemination. Going ahead, explain the process of spermatogenesis with this schematic representation. So over here you have spermatogonia, they increase the number, and now you have one cell, okay, one cell, primary spermatocyte. It will grow, and from the primary spermatocyte it will undergo meiosis 1. Your secondary spermatocyte, here primary spermatocyte has 2n number of chromosomes, here it has become n. Again it will divide, give you spermatid. From spermatid you get spermatozoa, that is known as sperms over here, but actually it is known as spermatozoa, and this process is known as spermiogenesis. At the end it moves out from the seminiferous tubule, which is known as spermiation. Done? Okay. And it happens at the puberty because GnRH, gonadotropin releasing hormone, its amount increases, and it releases LH and FSH. I already told you FSH goes and acts on fertility cells, LH goes and acts on Leydig cells and help in the production of testosterone. Now explain the structure of sperm with a neat label diagram. Over here you have the head region. In the head region, my dear children, you have acrosome at the end, which helps in having a hydrolytic enzyme needed for cleaving through the zona pellucida. Okay, here it is a nucleus, haploid nucleus. This is the middle piece. Here you have the neck. In the middle piece you have the mitochondria, which helps the tail to move. Whole of the thing is basically covered by plasma membrane. Going ahead, explain the process of oogenesis with the schematic representation. This happens in the fetal ovary only. Okay, when they are babies inside the womb, it happens. Over here again, oogonium is there. It undergoes mitosis, increases the number, and then one of them become the primary oocyte. Primary oocyte will be having 46 number of chromosome. It has not completed meiosis. Meiosis, when it is completed, my dear children, that's when you get secondary oocyte and also first polar body. So over here, this cell is not going to divide further because it has become a polar body. It will degenerate. Only one cell will go ahead with meiosis 2, and it will form ovum and second polar body. And my dear children, again you get one ovum from oogonia. Please remember that. Here you have the structures, okay. Primary oocyte basically have a primary follicle, so many granulosa cells, and then in secondary oocyte you will be having theca layer and the granulosa cell. In the tertiary follicle you will be having theca as well as basically antrum, okay, and then it will mature into Graafian follicle. Please remember that. Okay, in the tertiary follicle it will finally divide its completion and make secondary oocyte. Now the secondary oocyte is going to form a layer that is known as zona pellucida when it is in the Graafian follicle, okay, and also corona radiata is formed from the granulosa cell itself. Just have a normal thing. And when does the zona pellucida dissolute go away? After fertilization. After fertilization it goes away. Okay. Now list any 2 principles to prevent sexually transmitted diseases. Avoid sex with unknown partners. Always use condoms. Take advice of doctor for early detection and treatment. Now what is infertility? When the couples are unable to produce children in spite of what? Unprotected sexual intercourse. Okay. It can be due to congenital reasons, that is genetic reasons, physical, drugs, immunological, or even psychological. Going ahead, what is medical termination of pregnancy, MTP? Mention the safe period. Okay, medical termination means induced or intentional or voluntary termination of pregnancy before full term. Okay, and in other words, it is also known as induced abortion. MTP is considered relatively safe up to 12 weeks of pregnancy. But how far you can do? Up to 24 weeks you can do, but that's not safe. Up to 12 weeks it is considered to be safe period. Okay, going ahead, differentiate between spermatogenesis and oogenesis. It is process of formation of sperms. There you get egg. It takes place within the seminiferous tubules. There it takes place within the Graafian follicle. Oh, it's not like that. It takes place within the ovary. Okay, just that's enough. Stage of growth is short, stages of growth is long. It yields 4 functional sperm. At the end you get only 1 functional ovum. Polar bodies are not formed. Here polar bodies would be formed. Okay, and it starts at the puberty. It starts at the fetal stage only. Differentiate between sperm and ovum. They are produced in the testes, they are produced in the ovary, they are minute size, they are relatively large in size. Yes, the biggest cell in the human body, right? They are visually flagellated, they are non-flagellated, they are motile, they are non-motile. Okay, mention the techniques for infertility control art. Okay, you have in vitro fertilization, you have embryo transfer. Okay, after fertilization you can do embryo transfer, or probably you can just directly give gametes for the person who does not produce gamete, or probably you can just this comes under this only. Okay, basically you can just write in vitro fertilization. After that you can do ZIFT, that is zygote intrafallopian transfer. If it is 8 blastomere stage, if it is more than that, basically you have to do intrauterine transfer. Okay, intrauterine transfer, IUT. And then basically if you do not have gametes at all, then you do gift, okay, where you are gifting a gamete. Okay, otherwise if in the male they are not able to inseminate, you have artificial insemination. These are all parts of art. Going ahead, what is test cross? Test cross is basically you are going to cross a dominant phenotype with dominant phenotype, as in it can be this or this. So for knowing the genotype, you are going to cross it with what? You are going to cross it with a recessive parent. With a recessive parent. When you get unicross, you get to identify what is the actual genetic makeup. Okay, I have given you example. Please watch the videos if you're not able to recollect, or just use your NCERT as of now. Now going ahead, what are the conclusions drawn by T.H. Morgan from the crossing experiment in Drosophila with respect to linkage? When he crossed, he got to know about linkage, that is when the genes are closely associated, when you guys are best friends with each other, there is no chance for recombination. Closely associated genes, my dear children, will show very less amount of recombination. If the genes are far away from each other, they are going to show more amount of recombination. Please remember that T.H. Morgan was a person who termed the linkage term, and gene mapping. Okay, the genes are located this much, and thing like distance between the genes was given by his student, whose name was Alfred Seward. There were 3 guys who basically rediscovered Mendel's work. You remember them also: Schemark, Correns, and De Vries. Okay, why did Mendel select Drosophila? Because it can be grown easily in the laboratory. The life cycle is very small. Okay, single mating could produce multiple progenies, and they have clear differentiation of male and female flies. They have many types of hereditary variation which can be easily visible under low power microscope. Now inheritance of one gene, my dear children, show everything. Okay, take capital T capital pure breed, put the gametes over there, put F1 generation over there, make the Punnett square and write it like this. Okay, in F1 generation all of them are going to show the dominant phenotype only. In the F2 generation, my dear children, they are going to show, okay, 3 of them are going to be tall and 1 of them is going to be dwarf. Okay, please remember 3:1 ratio is the phenotypic ratio. The genotypic ratio is 1:2:1. Now Mendel's dihybrid cross, again start with F1 generation. F1 generation, all of them will be showing the parental trait that is round and yellow. Okay, in F2 generation there will be a ratio that is 9:3:3:1, where 9 is the dominant parent type, that is round and yellow. 1 is the recessive parent type, that is wrinkled and green. And over here you have to make new combination. Round was with yellow, over here you make round with green. Here wrinkled was with yellow, wrinkled was with green. Here you make it with yellow. Then 3 + 3 = 6. 6 recombinants are found. 6 new type of organisms are being found. Okay, what is the number of phenotypes over here? If you are asked 1, 2, 3, 4 phenotypes you can observe. What are the number of genotypes over here? 9 genotypes will be observed. 9 different type of genotypes will be seen over here. Okay, and which law does it show? Law of independent assortment. Law of independent assortment. Going ahead, Incomplete dominance. Incomplete dominance where it is not completely dominant. Example is 4 o'clock plank or snapdragon or basically mirabilis dilapa. Same name. Okay. So R, that is capital R, is not completely dominant over the small r, which is why you see pink color clouds. In the F1, F2 generation also you find 1:2:1 ratio, both genotypic and phenotypic ratio. Okay. Same. Now going ahead, you have observation. Okay, write in the 4 symptoms of Down syndrome, my dear children. The symptoms are short-stature, large head, small nose, and also open mouth with protruding tongue. Eyes. You don't need to write all this thing. Okay, they are mentally retarded, and also there will be palm crease. There will be palm crease, visible palm crease that will be observed over here. Okay, it is, please remember, Down syndrome is trisomy of 21st chromosome. Trisomy of 21st chromosome. Okay, it is aneuploidy thing that you can see. Aneuploidy means gain or loss of not electrons, chromosomes. So over here you see a gain of chromosome, my dear children, that is trisomy of 21st chromosome. How do you write it? Basically 45A plus XX or 45A plus XY. So we usually have 44 autosomes, but over here 45. One extra is there, 21st chromosome is the extra one. Now going ahead, that is a karyotype. That is the karyotype. Now write the symptoms of Klinefelter syndrome: abnormal body proportion, tall stature, diminished pubic axillary facial hair, enlarged breast tissue, that is gynecomastia, and small firm testicles. Okay, they do not show sperm formation. They do not show sperm formation. Over here they will be having feminine body. Okay, the feminine body will be there. Now, mechanism of sex determination in the case of different organism. XO type of sex determination in insects, for example, grasshoppers. Okay, the male are heterogametic, that is XO. They are producing 2 type of gametes, X and O. X and O as in there is nothing being produced in one gamete. Okay, X and XY, that is in Drosophila and human beings, ZZ and ZW in the females, in the birds, my dear children, and the female is heterogametic over here. And there is haplodiploid sex determination found in honeybees, where diploid is female and the haploid is male. Going ahead, what are multiple alleles? Multiple alleles where multiple alleles are there for one particular gene itself. Please know about pleiotropy. Pleiotropy is basically when one gene is affecting multiple traits. Example is phenylketonuria. There, mental retardation and also skin color, reduced skin color pigmentation. And polygyny where multiple genes are there for one particular trait. Example is skin color. Okay, just go through it once. Write a note on Mendelian disorder. So you have hemophilia, my dear children. Hemophilia is basically when a protein, that is a cascade of protein, is affected and it is going to affect the clotting. Okay, it is a sex-linked recessive disease. The males are prone to have this disease more. Okay, and the females can be a carrier if they have one chromosome. Now, males, if they have one chromosome, they are going to be affected. Now, you have sickle cell anemia. Sickle cell anemia is basically substitution of glutamic acid by valine. So, always remember it was GAG. A comes before U, right? Then later it is converted to GUG. On the 6th codon, this particular change is going to happen. On the 6th codon, my dear children, this particular change is going to happen. Always remember that glutamic acid was the first thing that was being produced, but now it is valine. And sickle cell anemia is a qualitative disorder. The quality of beta globin chain is going to be disrupted, and always remember the beta globin chain, this particular gene is located on chromosome number 16. Chromosome number 11. Chromosome number 11. Okay. And it is a autosomal recessive disorder. Anyone can have it, male or female, anything. Okay, phenylketonuria, again autosomal recessive trait. Over here, what happens? Phenylalanine hydroxylase enzyme is not being produced, which is why phenylalanine stays in our body and converts to phenylpyruvic acid, causes mental retardation and everything. Usually, phenylalanine transport is basically converted to tyrosine. That is not being seen over here. So, it is accumulation of all this thing. And in the urine also you will finding crystals and stuff like that. Always remember color blindness is also there, which is not being mentioned over here. Color blindness also had the same thing, just that the rod cells and the cone cells are being affected in color blindness and you won't be able to see any color out there. Okay, especially the red and the blue. Now going ahead, we have and there is one more thing, thalassemia, my dear children. Thalassemia is where alpha and beta both can happen, and it is a quantitative blood disorder. This is the qualitative one, that is the quantitative one. Okay, chromosome number 16th is of alpha and chromosome number 11 is of beta. Always remember that. Okay, write a normal chromosomal disorder, Down syndrome. I told you already. You can write this, Klinefelter's. So 47. That in Klinefelter's, my dear children, what happens is that when one extra chromosome is there, that is X chromosome. So you get 44, that is autosomes, plus XXY, that is 47. Okay, and whatever happens, overall masculine development, also develop feminine character, that is gynecomastia and everything. Now Turner syndrome, you have absence of one X chromosome. So over here it will be XO, there is no other chromosome. So these females are going to be sterile with rudimentary ovaries. Their ovaries are not going to work properly, which is why there is no ova. They lack secondary sexual characters and they have masculine body as well. Going ahead, mentioned in 3 applications of DNA fingerprinting technique. So an individual's DNA shows polymorphism. Each of our DNA are kind of different, and if we study on the basis of pattern of that, we would be able to identify a lot of things because we are not common. Okay, so It is used in the forensic lab. It is used for paternity testing and solving the disputes, determine the population and genetic diversities. Okay, it is there for breeding patterns to save the animal facing the dangers. You don't need to do all this thing. At least these two things are very, very important. Going ahead, list the salient features of Human Genome Project. So, human genome consists of 3164.7 million nucleotide bases. My dear children, 300 bases are there in average gene, but you have multiple things, dystrophin with 2.4 millions and other things. Okay, just go through it once. I'm not going to the entire thing. Chromosome 1 has the most genes and chromosome Y has the fewest genes. Okay, scientists have located what SNPs, that is single nuclear polymorphism, where only we differ from only single nucleotide. Okay, all and functions of almost 50% of discovered genes are unknown, my dear children. Less than 2% of the genome actually codes for protein. Rest all of it is repetitive sequences. Okay, describe the lac operon, very, very important. So you will be having ZYA, Z codes for beta carotidase, Y codes for permease, and A codes for transacetylase. They have a promoter region, they have an operator region. Apart from that, there is a regulatory region which codes for repressor protein. This repressor protein binds on the operator region, and what will happen basically, RNA polymerase would not be able to transcribe all this thing. When lactose or allolactose is there, it goes and binds with this, and hence it won't be able to bind over here, since transformation can happen then. Okay, transcription can happen then. Okay, beta galactosidase will be able to break down lactose to glucose and galactosidase, and permease will be helping in the entry of the lactose, and transacetylase is there for dealing with all the middle things that will be formed. Okay, going ahead, DNA replication, my dear children, this is the diagram. Always remember from 5' to 3' direction, and this is from 3' the opposite direction. Okay, always remember the one that is in 3' to 5' direction, it will be undergoing normal replication, my dear children, normal replication. So over here it is leading strand, but over here this is lagging strand, because what happens is that DNA polymerase needs primer. So every time it has to attach primer, which is why there are broken fragments which are known as Okazaki fragments, and they join together with the help of DNA ligase. For this you need DNA polymerase. Apart from that, you need nucleotides also. Nucleotides, my dear children, that is deoxyribonucleotide triphosphates, are going to act as energy source, and also they are going to help as a substrate for the whole process of thing, and it needs origin of replication as well for to begin things. Yeah, Griffith principle, please remember that you need Streptococcus pneumoniae for transforming the bacteria, my dear children. The S strain is the virulent one, the R strain is the non-virulent one, and once when you are putting basically the heat killed S strain and the R strain, what happens is that it is transforming this thing. Okay, but there was no one who was able to prove it until and unless Avery, MacLeod and McCarthy came in. They gave a proof for that. They used DNase. When the DNase was used, basically transformation was not happening. Still people didn't believe it, so a next person came who was Hershey and Chase. Okay, they did the experiment with the help of bacteriophage and the E. coli, where they use S35 radioactive thing that is going to label the protein, and P32 for labeling the DNA, and they found that basically the DNA is the genetic material. Just go through it if you want to have a look. Translation, my dear children, first thing is activation of amino acid, or charging of tRNA. On the tRNA, when the amino acid come and binds, you call this as charging of tRNA. Very, very important thing. Okay, and after which what happens is that the mRNA will be binding to the small subunit. Okay, it will be binding to the small subunit, and on this mRNA and the small subunit, tRNA will be coming, and slowly large subunit will be coming. The whole thing is being formed. Okay, large subunit attached. Okay, then initiation happens, and after that elongation happens. Larger subunit attached to the initiation complex. Larger subunit has two sites, A and P sites. You don't need to write all this thing. Okay, formation of peptide bond occurs. So one tRNA, another tRNA carrying two amino acid. The formation of peptide bond occurs with the help of 23 srRNA in the bacteria. They move from 5' to 3' direction. Always remember, RNA polymerase, DNA polymerase, as well as the ribosome are going to move from 5' to 3' direction, my dear children. Okay. Termination is when a row factor will be coming and attached, because there is no amino acid for the stop codon, right? So row factor will be coming and attaching and doing the termination. Always remember, you have to know the sequence of everything. Okay, you have to know the sequence of lot of things. That is RNA polymerase 1, what does it do? RNA polymerase 1 makes the rRNA, ribosomal RNA. RNA polymerase 2 helps in transcribing hnRNA, that helps later to form the mRNA. It has to undergo capping, splicing, and trailing as well. These are known as processing. Okay, what do you mean by splicing? You should know the removal of introns. What is this structure? This is nucleosome. Okay, nucleosome has 8 histone proteins, octamer of histone. Apart from that, DNA is wrapped around it twice. Okay, and there is H1 histone as well. one more very important thing is that basically there is euchromatin and heterochromatin. If they are very close to each other, okay, this is known as what do you call it? Say, what do you call it? You call this as euchromatin. If they are very loose, you call this, sorry, when they are very loose, you call it euchromatin, and the other one, when it is very tight, you call it heterochromatin, my dear children. Euchromatin is transcription active, heterochromatin is not. Going ahead, what is transcription? Again the same thing, you have initiation, elongation, everything. Remember, that is a sigma factor that comes and attached first for initiation, and then in the promoter region, okay, and then elongation it goes on, okay, that is RNA polymerase makes the RNA, and then in termination the rho factor will be coming and attaching. It occurs in 3 different steps. It is very, 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 very important. Over here you have to remember the transcription unit and everything. Please make a transcription unit if necessary. Now, what are salient features of genetic code? It is unambiguous. That means one codon is going to code for one amino acid. Degeneracy, some amino acids are coded by multiple codons. There is comaless, it is universal, and except some, yeah. There is stop codon, that is UAG, UA and UGA. Okay, so UGA, UAG, UAA are stop codons. Apart from that, start codon is AUG, which has dual function. It codes for methionine as well as this. Codon is triplet, my dear children. There are 64 codons, 61 codes for amino acids. 3 of them are stop codons. Okay, all of this thing you should be knowing. They are non-overlapping and they are nonsense codon as well. Now, write a short note on. We already discussed this. Name the causative organism of the following disease. Pneumonia is caused by Streptococcus pneumoniae, common cold, rhinovirus. Okay, typhoid, salmonella typhi, ascaris lumbricoides, filarius, Wuchereria, Mancrotia, Wuchereria, malaria, Plasmodium vivax, Plasmodium falciparum. It is spread. Okay, vector, you have 3. Okay, one is malaria, that is caused by Anopheles mosquito, dengue and chikungunya by Aedes mosquito, and Wuchereria, that is filariasis, by Culex mosquito. Okay, Wuchereria is going to affect the lymphatic vessels, ascaris is going to affect the intestine as well. Have that understanding. And pneumonia is caused by Haemophilus influenzae as well. Going ahead, what are interferons? Interferons are released from a virus infected cell to the normal cell so that it can be safe. Now, name the diagnostic tool for detect AIDS. It is enzyme linked immunosorbent assay, my dear children. Okay, name the disease diagnosed for Vidal, that is, why do we use Vidal test? For typhoid. Right, any 2 methods of transmission of HIV, so sexual contact with infected person, by transmission of contaminated blood and blood products, by sharing infected needles, and in the case of intravenous drug abusers, and from infected mother to her children through a child by the placenta as well. Okay. Now, life cycle of HIV, very, very important. The genome is this capsid and the RNA. It has an RNA, my dear children. The RNA comes in with the help of reverse transcriptase. The RNA is made to DNA. The DNA later gets incorporated over here, and later it will be forming what? Multiple. It will be having RNA. It will be forming the proteins. Everything. It will be released. So this cell is macrophage. It is known as HIV factory. And where is it going to affect? Actually, the HIV actually affects the T helper cells, which are the backbone of our immune system, my dear children. Always remember T helper cells. Okay, and yes, that's all. It is going to make us very, very, very weak. This particular picture is very, very important. Go through it one by one. A, B, C, they can give you like that, and you have to label it. Now, draw a neatly labeled diagram of antibody molecule, 2 heavy chains, 2 light chains, 2 variable regions, 2 constant regions, my dear children. They have disulfide bonds, many of them, they just have one of them. This is antigen binding site as well. Going ahead, we have 5 types of antibodies, IgG, A, M, E, D. G is from placenta, it's passed through placenta, gamed, okay, gamed. Why is it not coming? G is passed through placenta, A basically is passed through colostrum, that is the milk, M and D is not mentioned, E is there in allergy, okay. Mention any 3 characteristics of cancer cell. Cancer cell appear to have lost the property of contact inhibition, no contact inhibition, and basically they grow very rapidly, invading and damaging the surrounding normal tissues. They reach distant sites through the blood, and this property is known as metastasis as well. Okay, and please, there are 3 carcinogens: physical, biological, and chemical. Okay, chemical is tobacco, physical as in UV rays, X-rays and things like that, ionizing and non-ionizing radiation, and then you have this one, biological as proto-oncogenes. So we have normal genes in our body. If they get mutated, it becomes cancerous. Or we have oncogenic viruses that transfers oncogene into us. Okay, that is also there. Okay, and in the techniques, diagnostic tools, you have biopsy, histopathology, CT scan and MRI. Okay, CT scan and MRI basically are used for internal organs, for seeing the internal organs if they have any problem. Apart from it, what do we have? Apart from it, You have antibody testing. Cancer patients especially produces certain type of antibodies, you can test that, okay. And also microbiological techniques are there, okay, molecular biology techniques are there, where basically what do? You see whether a person is having a chance of getting a disease because of the genes that they have. Now we have innate and acquired immunity. Innate, that is present from birth. Acquired, as it you get it from let's say throughout your lifetime, either by occurring the disease or basically infecting by getting an infection or by vaccination or things like that, okay. It is non-specific, that is very specific. It is inheritable and remains throughout the life. It is non-inheritable and lasts for a very short period. Now differentiate between active and passive. It provides lifelong immunity, it provides for temporary immunity. Here your body produces antibodies, here you're getting ready-made antibodies. This is naturally acquired immunity in the host in response to an antigen. This is ready-made antibodies are given. It is very slow and takes time to response. It shows an immediate response, my dear children. Example, you can give vaccination, immunization. Now differentiate between benign and malignant tumor. They are localized. Benign tumors do not move around. They are not localized. Growth is expansive. Here it is invasive. You don't need too much. There is no metastasis. There is metastasis. Now drugs and alcohol abuse, opioids, cannabinoids, coca alkaloids, hallucinogens and tobacco and everything. Please remember, opioids you get it from papaver somniferum. Example, heroin and morphine. Morphine is a sedative and a painkiller. Heroin is a depressant. It affects basically your CNS or gastrointestinal tract as well. And heroin is known as smack also. Picture of morphine is given in your NCERT. Please go through it. Cannabinoids you are obtaining from cannabis sativa and it is going to affect your cardiovascular system, my dear children. Example is charas, ganja, everything up there. Again a picture is given with a bridge like this. Okay, this is what cannabis. Okay, now coca alkaloids or cocaine, you call it coke or crack also. You get it from erythroxylum coca, which is of America. Which part of America? I don't know, south. I think. Just check it once, okay. And basically it is the one that affects your CNS again, okay. Dopamine transport, you will get a euphoria or hallucination. Apart from that, there are other hallucinogens such as you have belladonna, atropobelladonna and datura also, okay. And always remember, the sports people are abusing one particular drug and that is cannabinoids. They are using cannabinoids, okay, very much, okay, sports people. And apart from that, tobacco is there. Tobacco has nicotine. Nicotine affects the adrenal glands and hence adrenaline is released, which increases our blood pressure and everything, okay. Works. Now the time is up. Adolescence and drug abuse, everything is given. Just go through it once, okay. Explain the role of microbes in industrial. So you have multiple things, my dear children. Saccharomyces cerevisiae, brewers, yeast. Wine and beer are produced without distillation. Whiskey and brandy and rum are produced with distillation, my dear children. Yeast is there. Antibiotics, that is penicillin, my dear children. Okay, it is produced by Penicillium notatum, which is a fungus. And the experiment Alexander Fleming was doing was on Staphylococci, which is a bacteria. And 3 people got award for it, okay. The person who discovered this, Alexander Fleming, 3 people got because they used in World War II. That is happening in the US, yeah. And basically it was Ernstein and Howard Florey, okay, and also Alexander Fleming, who got the Nobel Prize. Chemicals and bioactive molecules, very much important. Cyclosporin A, which is an immunosuppressant, you get it from Trichoderma polysporum. Streptokinase, you get it from Streptococcus, it is a clotbuster. And statin, which is a blood cholesterol lowering agent, you get it from Monascus purpureus. Apart from that, you have Aspergillus niger, which produces citric acid. It's a fungus, okay. Acetobacter aceti, acetic acid, Clostridium botulinum, butyric acid. Lactobacillus produces lactic acid. Saccharomyces cerevisiae, my dear children, production of ethanol. Please know which of them are industrial. Please know which of them are used to a home as well. So lactobacillus is something that produces a home as well, apart from here. Get it? Yeast also. Now write a note on microbes as biofertilizers. So you know bacteria, you have Rhizobium, Azotobacter and Azoospermilla, all of them fixing nitrogen, and this is found in leguminous plants. Cyanobacteria is a separate category. Blue-green algae is there. Anabaena, nostoc, oscillatory is there. It fixes nitrogen in rice fields and other aquatic places as well. Okay, fungi, you have mycorrhiza, my dear children, and it belongs to glomus. Very, very important. From symbiotic association with plant nodes, everything it helps in. It helps in phosphorus. Please remember, mycorrhiza, the fungi, is helping in absorption of phosphorus. Okay, this is the bioglass plant, very much important. Who put down this KVIC and things like that? Okay, very important. This diagram is also important. Over here, anaerobic bacteria are used always to produce biogas. Biogas is not just methane, it is also H2S and carbon dioxide. Going ahead, draw a neatly labeled diagram of gel electrophoresis. This is the gel electrophoresis chamber. You have wells and things like that. You have cathode over here and anode over here. DNA is negatively charged, my dear children. So it will be moving towards the positive end, that is the anode. Now, structure of PBr 322, very much important. Ampicillin resistant, tetracycline resistant. There are 1, 2 and 3, 4, 5, 6, 7, 8 restriction sites as well. Okay, in ORI there is nothing, but in ROP there is 1. Here there is 2, here there is 2, here there is 3. You should be knowing, they will be asking you to label it as well. This is simple stirred tank bioreactor. This is, this is sparge stirred tank bioreactor. Okay, just go through the diagrams once. Sewage treatment, you have primary treatment and secondary treatment. Primary treatment, you do filtration and sedimentation. After that, you have primary effluent, you put it in aeration tank. You give aerobic bacteria to grow. Aerobic bacteria goes and forms with fungal filaments. They will form flocks and they will require high amount of oxygen, that is BOD is way too much, and they break down the organic matter. If the amount of organic matter reduces, that means pollution reduces. That means BOD, the amount of oxygen also reduces, right? The need for oxygen also reduces. And later when the BOD is reduced, you put it down into a settling tank where the flocks and everything settle down as an activated sludge. Rest of the water is given to the river and things like that. This activated sludge can be used as little bit as inoculum. Okay, little bit of it can be put down into the primary aeration tank as an inoculum. Apart from it, everything will be put in anaerobic sludge digester where the anaerobic bacteria will be acting on it and producing biogas. Okay, you have Yamuna action plan and Ganga action plan on the basis of this. Differentiate between primary and secondary sewage treatment. These are the primary and secondary sewage treatment. Homeostasis is basically an ability to maintain what consistency in its internal environment. Whatever is the external environment, if you are able to maintain a constant internal environment, this is known as homeostasis. What are the causes of biodiversity losses? You have habitat loss and fragmentation, overexploitation, that is Stella sea cow is an example. Alien species invasion, you have Parthenium, Lantana and Ecornia. In the case of fishes, you have Claris garipinus, okay, that is the African catfish, and it was indigenous, was killing the indigenous catfishes. And then you have in Lake Victoria, you have this Nile perch, okay, killing the cyclid fishes which are there. Co-extinction, if one is extinct, the other one also becomes extinct. Biodiversity conservation in situ and ex situ conservation. In situ at its own place, national parks, sanctuaries and things like that are example. Ex situ are basically outside. So zoo, botanical gardens, meat, cryopreservation, seed preservation all comes under ex situ conservation. Here secret groups also come. Secret groups is very much important. What are biodiversity hotspots? A region where there is high amount of endemic species. That means endemic species means the ones that you cannot find elsewhere. Okay, Western Ghats and Himalayan region basically are major one. 34 biodiversity hotspots in the world. Indian subcontinent have 2 hotspots, Himalayan and the Western Ghats. Endemism, I told you, those are found in specific regions. In plant you have example Butea. In the case of animal you have Nilgiri langur. What are secret groups? These are the smallest patches that have protected trees and wildlife. Example, Khasi and Jainsheh hills in Meghalaya, Aravalli hills of Rajasthan, Western Ghats of Karnataka as well, and Chanda Bastar region of Madhya Pradesh. Very much important, okay? Sacred species, you have Ficus religiosa, that is people, Egle, Marmelos, that is bear, and animals like elephants and peacocks, okay? Important diagrams, my dear children, these are the important diagrams. Please make sure you are going through it. This also are important diagrams. And with this, we end it. Please remember, you have to go through RNA interference, very important thing. Okay, it is found in Meloidica and Incognitia. And what happens over there? It is going to affect tobacco plant. Okay, so we are silencing the mRNA by creating a double-stranded RNA to go and find it complementary, bind over it. Bt toxin are very much important. Okay, biocontrol agents, okay, all the biocontrol agents are equally important. Just go through it. Okay, I'll just name a few. Baculoviruses, then you have ladybird, you have grasshopper, sorry, not grasshopper, dragonfly, okay, and Bt, all this thing, okay. And cry genes, which cry genes are for what? Okay, I'll just say over here, cry 1AC and cry 2AB. Okay, so first letter is C, so it is cotton. Second letter is B, so it is bollworm. So cotton, bollworm, you have 2 genes, that is this. And for corn borers, you have cry 1AB. So C and B. There is only one word, that is corn borers. You can remember like this. I don't know how else to make you remember. Okay, just go through it, my dear children. Ayo. Yes, I'm stopping it right away. So it's already time has passed. So wishing you all the best and take care of yourself. You're going to ace the biology exam. Bye-bye. Take care.